Hello, everybody, and welcome to this ViewPathDB webinar. There's still some people joining, so we'll give people a couple of minutes to join. Please hang tight. I will go ahead and share my intro slide anyway to begin with. And I'll maybe cover some basics. Let's see, hopefully everybody can see my screen. Yes. Okay, so for GoToWebinar, you should have a control panel that allows you to ask questions. There's a question and answer or question panel. And so during the webinar, if you have questions or if you already have questions, feel free to type them there. And we have a number of people, as you can see from the intro screen. We have a few people from ViewPathDB who are on, who will be describing various features and new data in the latest release. But we will also be answering the questions as they come in. I think that's probably the best way to communicate with us regarding during the webinar. The webinar is being recorded, and so you will have access to it later on. So if we say something too quickly, or you're not sure about, or you have to leave early, or whatever, the webinar will be available. Everybody who attends the webinar will receive automatically, you'll receive a certificate for the webinar. So please check your spam folders. Those sometimes get filtered out. Of course, if you don't receive one, let us know and we can resend you one. But they go out automatically every day or every post webinar, I think 24 hours after the webinar. I think that's it. Yeah, that's all I was going to say here. So I think we can go ahead and start it. So that's 2 minutes after a thing. We have over close to 60 attendees, so that's good. I think that's a good number. We can go ahead and get started. So welcome again. So this webinar will cover release 50 of ViewPathDB databases. And this will cover the databases that received new data and also cover some new features and functionalities. So today presenting will be Evelina Besenko, who's based at the University of Liverpool, Gloria Giraldo Calderon, who's based at the University of Notre Dame, myself, Omar Harb, I'm based at the University of Pennsylvania, and Suzanne Warrenfeldt, who's based at the University of Georgia. So all of our webinars, so I'm going to just take you to our web browser, and you can see ViewPathDB right here. This is the main site. You'll notice here there are various announcements. We put them up here. You can close these, which is a nice thing because they sometimes clutter up your page, but you can always get them back by clicking on this little microphone icon, and then they pop up again. But that's a nice way where you can customize your view. Under Help, you will see that there's a section called Learn How to Use ViewPathDB. And there are a couple of things here that I would like to point out. So one is that all of the webinars are posted in the webinar tab or button here. So if you click on Explore, this will give you a list of all the webinars, future webinars, and also past webinars. And all of the past webinars are linked to their recordings. So you can go and look at this long list of webinars here. And if you're interested in RNA sequence searches and you want to go into more detail, you can click here and then go to that specific webinar and learn more about it. So we keep everything available to you so you can access it later on. The other thing I wanted to point out is that, I mean, you'll notice here there's a lot of help. All of our workshops are posted as well. So if you want to really delve into more detail into how to use the resource, you can go to our workshops. For example, I'll just show you quickly one of our more recent workshops, and you'll see here all the exercises are linked, and each one of these gives you a PDF, and then you can walk through it step by step in excruciating detail, but obviously, hopefully very useful for you. And the final thing I want to point out about this help page is that there is a calendar here that shows events from the NIAID, so the National Institute of Allergy and Infectious Diseases, which actually funds ViewPathDB and its sister BRC, which supports bacteria and viruses. So there are events both by ViewPathDB and also Viper, Patrick, and IRD. Those are listed in this calendar here. So you can go through and see. So if you're interested in bacteria, for example, and let's say vectors, you can obviously, you may be interested in looking at what are the other events from the other BRC. And so that's a nice way for you to follow up. And obviously, if you subscribe to Google Calendars, or if you can, depending on where you are in the world, you can subscribe to this calendar and follow it. Okay, so without delaying this much longer, I will turn this over to Gloria, who is going to describe some new data sets in Vectorbase. Gloria, I'm going to make you the presenter. All right.
Thanks, Omar. Right. Can you see my screen?
Yes, we can. Mm-hmm.
Okay, hello, everybody. Today, I'm going to talk to you about 3 things that make part of the Vectorbase release. So those are RNA-seq, ChIP-seq, and population biology data. But before I give you more details about the RNA-seq data, I'm going to explain how we integrate the RNA-seq data in general for the ViewPath resources. So this is the page that Omar already showed you, which he said could be accessed from help, learn how to use ViewPath. And if you click here in methods, you will be able to see how exactly is that we incorporate and process the RNA-seq data and the ChIP-seq data. In the bullets, you will find an overview of the steps, but if you really want to know the details, you can click here and this Google document will give you all the details that you need. So important to mention that we curate the literature to include all the data sets that we have available in the web page, but we are always open to hear from you from data set that either you produce or you are interested from another lab. It could be or have already been published or you are about to be published. But the most important thing is that you need to submit that data to any of the INSDC's archival databases. So we directly pull the data from SRA or ENAP, so you don't have to submit twice. You just let us know what is your accession number, and we will incorporate the data. How often do we update this? Every 2 months. Where are you going to see this data? So we can incorporate the data, for example, for RNA-seq in the genome browser, and you go to tools, genome browser, that's one of the options to visualize the data. We also have the RNA-seq data available in Apollo. This is our annotation tool. So when you click here and go Apollo, you will see this screen, and this tool will give you RNA-seq tracks for you to use as evidence to annotate genes. And the one that I'm going to explain today in the detail is the search strategy. So this search strategy is available both from here, searches and search for. And you have different names, but they do actually exactly the same thing. So if you start typing here the word RNA, notice how autocomplete suggested me these 3 options, and I select one. This is the one I want, transcriptomics RNA-seq evidence. And if I click here, this is, oh, sorry, I forgot to mention this. I went ahead of myself. So if you go here to data sets in Vectorbase, you will have a list of all the data sets that are available. And if you want to know only the RNA-seq data sets, you click on category and type RNA-seq, and you will know. Out of the 481 data sets in Vectorbase, 322 are for RNA-seq category, but not all of them are available in all the tools, and that's what I'm going to show you now. Okay, so I type RNA, have my 3 options, and I land here in this page. So in this page, you can filter by organism alphabetically. And here, this is not the titles of the papers. This is the title that we give, because if you click here in question mark, you actually get the paper and a summary that we write. But sometimes the papers have more than one experiments or different things, not only RNA-seq. So we provide our own title and we provide the author. So you can start typing keywords like I did here. So for example, you want studies related with blood meal or with sugar meals, or like I had here for females, for example. And notice that for Aedes aegypti, there are about like 8 or 10 studies related with females. And I'm interested in this one. I read the summary and I said, oh, this is really what I want to do, is comparing these females that had been fed sugar or blood. And I have different options at the end. So notice is differential expression, fold change, percentile, sense, and anti-sense. So when you hover with the mouse, it will give you a description. But what these do is for differential expression, you can compare two different samples and give you statistical significance. But if you do just the fold change, you can still compare two different samples, but it will give you just like a mathematical difference without the statistical support. So let's say I want Bonisoni study and I select differential expression. Once you get here, so you have your experiment, you make sure that's the one you want. You select the reference sample. So I want my reference sample to be sugar, and I am going to compare this to the blood sample. Here, what direction do you want to compare? I only want the upregulated genes, and by default, it gives you a twofold difference, but you can change this to anything you want. And of course, you can also change your p-value, and you click on Get Answer. Once you do that, it will load for you this page in which you can start to grow your strategy. So in this case, for this experiment, I got more than 1,000 genes, but you can revise that strategy and change the parameters, or you can add more steps and include your list of genes. And here at the bottom is the results, and you can add more columns to see more details about this. We had a specific webinar about this, and this webinar I'm going to post in the chat box the link, so you can get into more details how to use the RNA-seq data and how to grow your strategy. So now that I said that, so you can better understand this bullet that we had here. We have 327 RNA-seq data sets, but for the strategy tool, only 113 of them are available. But in every release, we make more and more available, so expect to have more in the search strategy. The other thing that I wanted to say is that also here in the release notes, we have chip sequencing data, and that data can be visualized in the genome browser. So you just need to make sure is the appropriate species. So here you can see is Aedes aegypti, and you come, select the track, and I just start writing the word chip, and I get these options here. These are the tracks that I want, back to browser, and you start visualizing the data. How I'm doing on time? Can I have some time to real quick show the PopBio data?
Yes, Gloria, you can do another like 3 minutes. You should be fine.
Okay, thank you. So here it is. Okay, so the other data that we have for this release is population biology data. And let me load this here. Okay, this tool is currently only available for vector-based, but it will be available for others or of the sites as well. So you go to Tools and click on Map View, and that's how you access. And as you can see, we have data worldwide and different types of data. I'm going to move this here. Okay, so what are the types of data that we have? For example, abundance, which is how many mosquitoes or other type of arthropods are collected in an area. Or we have, for example, pathogen. What are those vectors infected with? Plasmodium, West Nile virus, or other arboviruses. Or we also have blood meal hosts, for example. What are the source of the blood? Did the vector bite a human, a cow, a rabbit, a pig, or a dog? Or we also have insecticide resistance phenotypes in which arthropods are brought to the lab and they are tested. So for this release, if we go back to the release notes, we have all these new sets available. And here we have population abundance data for the state of Montana. And we also have other types of studies. So if I go here, so these are the, you can search these here in the search box with a project ID, which people publish in their papers, or you can just type a keyword. You say, I don't want that specific paper. I want, that's like the section number from GeneVac. I want all the data from Montana. And that's how you will obtain it. And when you select a marker, you have different options. So for example, here I select one marker, and if I go to this plot here, I can see over the years, what is the different distribution of the species. When I select, I only want to look at population abundance. Or for example, I can select a marker and I click here in summary of a species, and I can see what are the most abundant species. Or well, here's this insecticide resistance. I can see which insecticides have been used, and I see there have been used 3 different insecticides with this sample, and in the outer ring show me the species, and inside is orange, so it's towards getting resistance here. And also the metadata. So for each sample, you're gonna get the details about how it was collected and what details we have. And the last thing I want to say is that you can download this data. Our curators spend a lot of time making everything standardized. So from every single data set worldwide, you can make your queries and browse, use date search or seasonal search. I only want to look at the rainy season or the dry season for a specific data set. And when you download the raw data, please remember that, you know, cite the source where you take the data from. And that's all I have to say. Hope I didn't went over my time.
No, that's good. Thank you, Gloria. And as a reminder for everybody, feel free to ask questions in the question and answer panel, and we will answer them either verbally or by typing. So I'm going to take a few minutes and cover some new features and data sets in ToxoDB. So hopefully, you know, you're appreciating that all of the ViewPathDB databases are built on the same infrastructure. They all look the same. You know, the small differences between them will include, you know, the type of data that's available for a particular organism. But in general, all the searches are available across all of these databases. And an important feature of ViewPathDB databases, in case you're not aware of this so far, is that we're not really a repository of the data. We are not the place where people store the data, but rather we're a place where scientists like you can come in and start asking questions across different data types. So a data set may be stored in, a proteomics data set may be stored in PRIDE, you know, which is a repository for proteomics data, and another RNA-seq data set may be stored in the SRA and in GenBank. But in ViewPathDB, you would be able to ask, you know, questions like, show me all the genes that are upregulated at this stage or that stage, and also have proteomics evidence from a particular experiment. And so that's a nice way where you can now leverage these search strategies that Gloria was showing you to combine the knowledge available on the various organisms and from the various data types. So looking in ToxoDB, I'm just going to click here on the right-hand side where it says News and Tweets, and I'm going to select the ToxoDB Release 50 News, and this will show you what's new in ToxoDB. And there are 4 new things that I wanted to highlight, and I'll spend, depending on the time, I might spend more or less time on each one of them. We've integrated a new annotated genome, which is a PacBio assembly for an RH88 strain. This was done by the JCVI, but it was an NIH-funded genome sequencing center. So what's cool about this genome, I mean, it's yet another Toxoplasma genome and annotation, but what's cool about this genome is that it is assembled using PacBio, and as if you're familiar with Toxoplasma, it has been accepted that Toxoplasma has 14 chromosomes until probably about half a year ago or so, when evidence came out that, or strong evidence came out that actually there are only 13 chromosomes, and this genome is assembled into 13 chromosomes. You can search this genome just like you normally do in any of our databases, and here's a list of all the searches that you can use to explore this genome. And what I wanted to show you here is just a genome browser view. So I'm looking at this RH88 strain. I'm looking at a chromosome. In this case, I can click here. I'm looking at chromosome 8, and chromosome 8, in fact, is now the merged chromosome. And if you look here, I'm showing synteny or collinearity between RH88 and the standard default ME49 reference strain for Toxoplasma. And you will see here that there are two segments. One is chromosome 7b, and the other one is chromosome 8. So chromosome 7b and chromosome 8 are actually should be merged into a single chromosome, which is now called chromosome 8. So, and again, I won't go through the details of how to search a genome. I think those are covered in other webinars. And obviously, if you have questions, we're more than happy to cover these later on. Another data set that was loaded was this proteomics, is the quantitative proteomics data set. And this data set looked at ectosomes and exosomes from Toxoplasma. So Toxoplasma, when it invades and sits in a parastarper's vacuole, it secretes all sorts of proteins and structures into the parastarper's vacuole. Some of them self-assemble into structures. Other ones are just free proteins, and then there are also exosomes. And what this group did, and the paper is linked here in the dataset description page, they took the exosomes and ectosomes from in vitro parasites. So they took the parasites out, and then they just let them sit in a particular solution. They got these secretions from the parasites, and then they did specific proteomics on them. They used quantitative proteomics, which then allows you to ask questions about this data, quantitative questions about this data. So I'm just scrolling down to the bottom of the page. Here's an example gene, but I'm going to go to the search for this. So this is a search where I'm going to compare, in this case, I'm going to compare ectosome to exosome to exosomes, and I'm going to ask for any gene that is upregulated by at least 1.5-fold in the ectosomes compared to the exosomes. And if I click on get answer, this will return, in this case, it returns 73 genes that met the criteria that I was interested in. And as Gloria indicated, you can modify these, play around with them, and so forth. And then you can go down and look at these genes and investigate them further. All right, so the next data set I'm going to describe, let me go back to the news. And so if you are a Toxoplasma researcher, this is a pretty cool data set. It's a genome-wide mapping of transcription initiation sites. This work was done in Sebastian Laredo's group, and let me just make sure, give a proper idea. So Benedict Marcus is the postdoc or student who did the work. He worked with us very closely to make sure this data is integrated. And in fact, this data is integrated pre-publication, which, as you know, happens quite frequently in our databases. Researchers are excited to provide their data to the community through ViewPathDB so that people can already start using it and not have to wait for a publication to come out. And so in this case, what they did is they specifically sequenced 5' ends of genes. And as you know, it's always hard to define where's the exact beginning of a gene, where's the exact 5' UTR of a gene. So this data is loaded as a genome browser track. And so there is a link to the genome browser, and I have it open here just to make it load faster. And you'll see here, I'm looking at a view of the ME49 genome. These are a bunch of genes in here. You'll notice these white pieces here are UTRs, untranslated regions, 5' and 3', that are part of the annotation. It doesn't mean they're correct, right? Those were based on RNA-seq data, maybe, and based on the RNA-seq data, you kind of guess where the RNA-seq data ends, and you say, well, that's where it is. The cool thing about this 5' specific data is that it allows you to see the exact location of the transcription start, right? So now I can look in here and I can say, okay, it looks like for this gene right here, which does not have a 5' UTR, as you can see, there's no white section here. I can go over here and you'll see there's the location of the transcription start based on this data from the Laredo lab. And you can now see the exact location of the transcription start. And in other cases, you can confirm, oh, it looks like the location, of this UTR is maybe correct. So we can zoom in a little bit here, and you can get a closer look, and then you can decide whether, and you'll see here that actually this UTR, the annotated one, is longer than what was found by the experiment. And so this is a really nice way for you to go in and really confirm whether your gene models are correct. So what's available here in the genome browser is what they determined based on all the analysis, which is shown below here. These are coverage plots of where the RNA-seq essentially aligned from the various experiments they did, both in tachyzoids and bradyzoids. And this is a strand-specific experiment, which is obviously very important if you want to do this correctly. But then they essentially collapsed the results into these defined start sites. And so you can see there are lots of peaks here going in, there's a peak here, but there are all other minor peaks here, but the largest peak is what is defining the location of the start site. I think this is all I'm going to say about this data, just to suffice it to say that it's super helpful and really allows you to define locations. I mean, you can see here's another example of a gene with a 5' UTR, which looks to be very short, and the actual potential start site is all the way up here, right? So if you're trying to clone this gene to include its UTR, so its natural state that may allow it to be properly expressed and stabilized, then you may want to actually clone all the way up to here in order to get this gene properly expressed. Okay, and if you, again, with our data set description page, there's a lot of information that helps you understand the data, hopefully. And so again, you can explore this further and see if, and obviously let us know if you have additional questions. The final thing I wanted to mention for ToxoDB is that we made some improvement on the hyperlapid global mapping of protein subcellular localization data. This data has been available, again, pre-publication for a while in ToxoDB. It's a really cool data set where they looked at subcellular localization of all proteins or most proteins and used statistical methods to look at clustering of proteins and their subcellular localization. Again, the data is fully described on the data set description page. You can get a lot of information. It's obviously since been published, and so you can actually go to the paper and look at it. And I do have a gene page open here. So we have a search available, which I'm happy to show you, but for the interest of time, I'm just going to a gene page. And on the gene page, you'll notice here there's the section of pages divided into different categories. And I went to the protein targeting and localization section. And you'll notice here there's the hyperlapid data is available here. I can look at the description, which is expandable, so you can get even more information about this data. Then scrolling down, there's a nice table here. It tells me that this gene is most likely in the microneme. Sorry, it has a probability of 1, which is obviously very high. And then you have this graph, which is really nice because it shows you not only the probability of its localization to the microneme in this case, but also all the other probabilities in the other organelles. Because sometimes, it's a probability of 1, so the likely it's going to be anywhere else is probably non-existent. But sometimes you can get probabilities that are in different organelles. So it's nice to see that for a gene, not only the maximum probability, but all the other probabilities. And so in this case, based on this data, you can be pretty confident that this gene is a microneme targeted gene. Okay, I think I will stop here. Again, as a reminder, if you have questions, feel free to add them in the question and answer panel. And I will, so there's a question, I see a question about whether this data, and again, I'm not sure which data this is. So for the hyperlapid.
He was asking about the transcription start sites. That's when that came up.
Okay.
Right.
So this data is only available in ToxoDB. It's just that's what they provided. And obviously, the nice thing about our resources is that as soon as somebody generates the data in another organism, we have all the tools available. And so all we do is we can load it in a much easier and more streamlined fashion. And so definitely, if, for example, in this case for Babesia, I know this is Hiba asking the question. So Hiba, if you end up doing the experiment and defining transcription start sites, or anybody else who's doing that in Babesia, definitely, as you know, let us know, and we will be excited to integrate that data into Pyroplasma DB. Okay, I'm going to switch over now to Suzanne, and I believe you are going to cover a couple of other databases.
Mm-hmm.
Let's see the presenter. All right.
Okay. Thanks. So I'll talk today about two data sets that are new in release 50. One of them is in HostDB and one is in PlasmoDB. HostDB is our database that supports the host portion of omics experiments run in mixed parasite and host systems. So for example, many studies infect cultured host cells with parasites, and then instead of isolating the parasites
Suzanne, sorry to interrupt. I'm not seeing your screen in case you were sharing already.
Oh no, what, yes, I am sharing. What do you see?
I see.
Nothing.
Hold on. Let me do one more thing here. Eve or Gloria, can you see Suzanne's screen?
No, I can't. Just a white screen that says release 50 webinar. Suzanne, there should be a pop-up in your desktop.
Yeah,
You
I
need to allow
clicked show my
presenter.
screen. I clicked that.
Here, let me
I will stop
make you.
showing.
Yeah, stop it. And I can also like switch you. Okay, now we see your screen. Perfect.
Yes.
Oh, okay, good.
There we go.
Sometimes you have to click it again.
Yes.
Okay, so where was I? Like some studies infect cultured host cells with parasites, and then, you know, instead of isolating the parasites for RNA-seq analysis, they use the entire culture and they sequence all RNA that they get out of that. So, and then during the analysis, the RNA-seq reads are aligned to the host genome and to the parasite genome. And when we get that type of data, we house the host portion of that data in HostDB. And that's the website that you're seeing here. Now, the experiment that I want to describe is a little bit unique in that it only has host data. Now, this study is from a 2020 publication from Anita Koshi's lab, and they are looking at toxoplasma-injected mouse neurons. So they have 3 sets of mice in their experiment. They have saline-infected, type 2 toxo-infected, and type 3 toxo-infected. Then they wait 21 days, and they isolate the brains. And then they collect 5 types of cells using laser capture microdissection. From the saline-injected mice, they get controls. And then from the toxo-injected mice, they get injected neurons, which they know are injected neurons because they have they're green with the GFP. And they get bystander cells, which are not green, but are within 80 to 20 microns of the green neurons. Then RNA sequencing, and we integrate that data into HostDB. So, and then we, you know, have the full scope of our tools available. The data is available on gene pages and some searches. So, so I'd like to go and demonstrate the search. I'm going to look for it here. I'll look for searches, genes, transcriptomics, and RNA sequence. And then if I scroll down, I can see this little new icon. And I'll go ahead and look for use our differentially expressed gene search. And I'll choose uninfected or control cells as the reference, and then type 2 TINs, which are toxoplasma-injected neurons. I'll look for upregulated genes, and I'll just leave these the same. Fold difference of 2 with an adjusted p-value of 0.01, and I click Get Answer. Whoa, I find 4,531 genes, and that seems like a lot to me. So we have this, there's a way to, instead of doing this whole search again, to go back and revise. So I'm going to click Revise and keep my samples the same, but look for genes that are differentially expressed with a fold difference of 40 and a p-value of 0.001. I'll go ahead and revise that. All right, so now I have 983 genes that are upregulated 40-fold in toxo and plasma injected neurons compared to normal neurons. Then, of course, you're in the strategy system, and so you have, you know, at least 100 other searches that you can intersect with this. And you also have our enrichment analyses that you can do. So I am going to demonstrate that one here, and I'll do a gene ontology enrichment. I'll look for gene ontology terms that are enhanced or enriched in this set of genes compared to the regular genome. So this is our tool. I'll choose the biological process ontology and use everything else as default. I'll choose a p-value cutoff of 0.05 for my enrichment analysis. I click submit, and
Suzanne, can you refresh your screen? It looked like it froze for me as well right now. I only see the menu, the searches menu from the top.
Goodness.
There might be some, I see your mouse now moving around, but I don't see the analysis section, basically, which is strange. You could stop the sharing and start the sharing again, and I'm sure we'll see the screen again. Yeah, there must be some connection dropping or something. That happens.
It's funny because y'all can hear my voice, right?
Yep, exactly. Yeah, that's true.
So do you see this now? We're waiting for the gene ontology enrichment.
Yeah, I don't see it, and still frozen here. So thanks, Grace, who's responding. Okay. So maybe
yeah,
stop
maybe we
sharing.
should just move on.
Okay, I can move on to, or actually we can switch to Eve, who will talk about FungiDB, and then if we have time in the end, we can come back to this and see if the connection is better, or one of us can show it, obviously. So. Okay,
I'm so sorry.
Yeah, no problem. That happens in this world.
Alrighty. Bye.
All right, sounds good. Let me know if you can see my screen.
Yes, I can see it.
All right, great. All right, so I will briefly introduce you to the updates in FungiDB. And as you have seen, various types of strategies are available on our sites. And the cool thing is that no matter whether you're in toxoplasma or vector-based, the idea is the same. Navigational principles are the same, and you can expand and build comprehensive strategies just the same way. So in the release 50, we have several data sets for various pathogens, and that includes Coccidioides species such as Basidiaceae and Immitis, and also Aspergillus pathogens, Clavatus fischeri, and Fumigatus, and they are known to cause pulmonary diseases in animals and humans. So I will briefly go over the Coccidioides data, and for those who are not familiar with this type of fungi, Coccidioides causes coccidiomycosis, and there are two species that are normally genetically and geographically different in sense where you can find them. For example, C. Basidiaceae clinical isolates that collected from Venezuela and North American populations are normally distinct, and they normally largely found in Arizona, Texas, Mexico, Central and South America, while Coccidioides immitis primarily was found in Central and Southern California, but recent reports indicate that it has been found in Washington State. And the cool thing that you can do with the SNP data is that you can identify SNPs in different species and then compare different SNPs and determine which genes they're mapping to. So for example, if I click on individual links here, I will be navigated to the dataset page. And as you have seen before, you can scroll down to the bottom of the page, investigate and determine what type of searches are available for each dataset. And in this case, I'll be interested in determining a SNPs based on differences within a group of isolates. And here I already opened that screen in advance. So here we see a data set that has been mapped to Stray and Silveira, and that data set is for Coccidioides pasadisi. And another data set that we have is actually aligned to Coccidioides immitis RS genome, and that's the data that comes from the Washington State CDC labs. And in order to determine SNPs within a group, I will leave the default parameters as is, and I'll say I want to determine any SNP that has a read frequency thresholds of 80 and percent of isolate with that base call is 20. And so when I click on Get Answer, the search will be deployed, and I will return essentially a result for that search. In this case, we have hundreds of thousands of SNPs that are mapped to individual genomes and found for each of the data sets. And I will go back to my main screen and show you that I have pre-run these strategies already. And here's my data set for the Washington State. Here's my data set for Caribbean areas. And what I have done, I went ahead and cross-referenced SNPs that were detected in these data sets to individual cross-reference with individual genes, and essentially mapped SNPs that are located within genes. And my results indicated that I have several thousand genes that return that fit my criteria. And the next thing that I wanted to do, I was curious whether I could compare the results between these two data sets. Obviously, these data are mapped to different species. And we have a very powerful tool. It's called a orthologous transformation that can help you to essentially convert genes from one species into another. And it's very easy to deploy. I'll demonstrate in one of the searches. For example, I'll click on Add Step and then go ahead and say Transform. And this is where you can transform genes by clicking on the orthologs button and essentially selecting the species into which you want to transform from the drop-down menu. So my idea was to compare essentially the SNPs that I have determined in immitis and bring in the data set from Caribe. And so, for example, once I have determined the C. immitis orthologs in C. pasadisi, I was able to create a nested strategy. So let's go ahead and actually take a look at how to create one. So at the moment, I have two different strategies. Both of these strategies now return the number of genes in the same species. I started here with C. immitis, transformed it into C. pasadisi, and here I returned the number of genes with SNPs and C. pasadisi. In order to compare these two, I'll add a step, and I said that I want to compare the results, but instead of running a new search, I would like to add an existing strategy. And since I'm interested in bringing in SNPs determined in the Caribbean data, I would click on the Caribbean data set, which is right here, and the search will refresh in a few seconds to show you that when the comparison is made, there are several thousand genes that are the same, meaning that there are SNPs detected in orthologs of pasadisi and C. immitis, but you can also determine some of the more unique genes by editing the Boolean operator selections. For example, you can determine which genes come specifically from the Washington State data and revising your search, or you can also switch it back and say, well, you know, I want to look actually at the genes that were brought in from the Caribbean data. And notice that the results for the number of genes changes here. And as you have seen before, you can also scroll down to the list of genes and navigate to individual gene pages to visualize the data. One more thing that I would like to show you for the SNP data is that in addition to gene results, you can also visualize individual SNPs and the coding potential within gene record pages. This is a gene record page for one of the genes in my data sets. This is for histone. And for example, if I navigate to the genetic variation section, it will bring me back to the gene model and also show me all of the SNPs that we have identified from all of the data that we have for this species, and individual colors will indicate whether it's synonymous, non-synonymous, nonsense, and other types of SNPs. Okay, and I still have a few minutes, so I'll quickly navigate back to the main screen and talk to you about proteomics data. So similarly, as I have just shown before, you can compare the results from these datasets by using orthologous transformation. What is really cool about this dataset is that the authors have used an activity-based protein profiling to identify unique or overexpressed genes in 3 species. And obviously the expectation would be that the species may behave slightly differently when they are grown in minimal media or when they're exposed to human serum containing media. To run a search, for example, you can also navigate to individual gene record pages or deploy a search from the top, just as Suzanne have shown you before, and view the type of data that is available for this type of data set. I have pre-run these searches, and I just want you to quickly demonstrate the results of the analysis that I have. So here is, for example, the results that I have run for Aspergillus fisheri proteomics dataset and also Aspergillus fumigatus. And similarly to the strategy above, I wanted to compare the data using Aspergillus fumigatus as a sort of the baseline genome. So what I have done, I have, and here I'm clicking on Edit to Revise to demo my selections of the search. What I have done, I selected the same criteria for both data sets, such as I have looked for upregulated genes in using reference samples as the data where no serum was added to the minimum media, and also comparing it to both data sets collected at 24 and 48 hours where human serum was added. And I generated these results for both species. And what I have done next, in order to compare the and determine not only genes that are sort of similar between the 2 species, I transformed Aspergillus fisheri results into orthologs and Aspergillus fumigatus 293, and then essentially brought in another step where my next search was to compare the results from this strategy to the results of my search in Aspergillus fumigatus. And what I have determined that there are 3 genes that were similar in their expression between these 2 proteomics datasets. But as you can imagine, you can also click on Edit button and said, well, determine only those that are unique for Aspergillus fisheri, since this is the closest relative to the pathogenic fungus Aspergillus fumigatus. And once you revise, you will notice that now I have 12 genes. And I think I'm almost out of time, but one thing that I wanted to show really quickly is that in addition to the types of the searches that we have shown, you can also visualize and enrich your data based on the GO enrichment analysis, metabolic pathway enrichment, or word enrichment tools. Okay. I think this is all I wanted to show. Suzanne, would you like to try again?
Actually, I'll take over because sorry about that. So just because I want to make sure I cover the orthology stuff and we have enough time, I did quickly do exactly what Suzanne did just to show. So let me show you quickly. So first of all, thanks Eve for a nice demonstration of how you can use the search strategy and combining different data from different organisms. And this orthology transform tool that she was using is incredibly powerful, and I would encourage everybody to explore it, because you can start asking questions even across different, seemingly diverse species, right? So you can, in ViewPath-DB, for example, ask a question in Plasmodium, or if you're interested in Babesia, for example, you can ask a question in Plasmodium, but then transform the results to Babesia genes, right? So if you're looking at, if, for example, there isn't a particular type of data in Babesia, because there are more researchers who work in Plasmodium, and there's obviously more data there and more maybe funding in that field, but then you can actually leverage that data using the orthology transform. It's obviously something you have to always confirm in the lab, but it gives you a nice lead and gives you some ideas about where to go with your experiments by leveraging information or knowledge from other organisms. So in the example that Suzanne showed in HostDB, she found these 983 genes that are upregulated in the injected cells, and she ran a Go enrichment analysis. So if you go under, if you click on the analyze results, let me click here. So if you click on analyze results, you'll get options here of different types of analyses. One of them is a Go enrichment. When you click on this, you can actually select the parameters of the Go enrichment. Eve showed this as well briefly, and then you can see this is the results. I pre-ran this while Eve was talking, but you can see down here, these are all the genes that are that were enriched in the results. You can look at the statistics associated with those enrichments, right? So if you look at the corrected p-values here, these are obviously exceedingly good. So indicating that these are groups of genes that are or Go terms that are enriched in your results. So clearly there is immune and defense responses that are enriched in these infected cells compared to the uninfected cells or injected cells, which makes sense. So there is a question from, quickly, I'm just looking at a question at the same time. And so there's a question from Maritza about whether we have a Leishmania DB. Actually, we have something better. We have a tri-trip DB, which is a database that includes not only Leishmania, but it includes Trypanosoma brucei, and also it includes various Leishmania species and T. cruzi. And so, you can always go to our about section on any of our sites. And if you click on ViewPath-DB sites, you will see all the sites that are available, and you can quickly go to tri-trip-DB, and the colors will be slightly different, the logo is different, but all the functionality is there. So you can run exactly the similar or same searches as before. So let me go back here to HostDB. And actually, I'm going to now switch to OrthoMCL. So the last topic we're going to cover is some updates and new features that come to you from the OrthoMCL databases, which is one of our databases. So the biggest change here is that the OrthoMCL database is using the new infrastructure that now you're used to with all of the ViewPath-DB sites. If you want to learn more about OrthoMCL, there are a number of tooltips available to you here right in the central portion. So you can click on each one of these and you'll get more information about how to use different features in OrthoMCL. So if you're curious as to, well, what is OrthoMCL to begin with, here's some information with links to publications. But in a very, very basic terms, OrthoMCL is a resource that uses a couple of different algorithms. It uses BLAST similarity and then uses the MCL clustering algorithm to put proteins together into groups, which we call orthology groups. So when Suzanne or Eve, or maybe it was just Eve, ran the orthology transform tool, right? The question she was asking is, show me the, basically in the background, it's saying, show me the group that these proteins belong to, the groups, and then from those groups, if there's a member from that group that is from the other species, give me that other species, right? Because they're in the same group, that means they're orthologs of each other. But basically, we're leveraging the orthology or OrthoMCL results. So there are a number of searches available to you in OrthoMCL that allow you to search both based on orthology groups or only based on the sequences. And OrthoMCL includes many hundreds of organisms across the tree of life, right? It's not only specific to parasites, but sorry, not the parasites, but not only specific to organisms that are in ViewPathDB sites, but it's highly relevant because when you are asking a question in, let's say, Plasmodium, you may be interested in knowing, well, which genes do not have orthologs in mammals, right? Or which genes have orthologs in plants but don't have orthologs in mammals, and that's a very important feature that leverages the orthology tool, which I will highlight in a second. So you can run this phyletic pattern search. You can run it in OrthoMCL if you're just interested in orthology and you're curious about ortholog groups that belong to different taxa. And you can see here, this is basically a tree, an expandable tree, and you can go in and look for your organism of choice, or you can quickly start typing in here, let's say Falciparum, and you'll see these are all the Falciparum genomes that are available in here. I can select, let's say you're interested in genes that are conserved between Plasmodium falciparum. I'm going to pick Babesia. Let's take Babesia bovis, and let's pick Toxoplasma. Toxoplasma. And so again, I'm just picking this randomly. So I'm asking for genes that are conserved across these 3 different organism groups. And you'll see here it came back with 1,766 orthology groups, right? That include proteins from the 3, taxa I selected, and obviously it'll include other things because I did not limit the other organisms. And then it'll give you basically each group with the number of proteins in their group and a phyletic distribution of what phyla are present in this group. So you'll see that the alveolata are there. I selected alveolates anyway, so that's great that they're there. But you also see that there are other organisms represented in these groups. I did not limit those, and so that's why you will see them there. So the strategy system works exactly the same as in all of our sites. There are a number of tools available. One tool that's very popular, this is this assign your proteome to orthology groups. This is used by people who annotate newly sequenced genomes. And this is a Galaxy pipeline. We generated the Galaxy, we put the tools into Galaxy, so it makes it easier to run. But you can go to Galaxy, you would have to log in and have an account because you're using storage space. But ultimately, you'll be able to upload your own novel transcript or proteome that you translate in silico if you're, for example, interested in annotating a new genome, and you'll do this by virtue of the orthology groups they belong to. And this is quite heavily used. I won't demo it right now just for the sake of time. But I did want to hop back to, let's go back to ToxoDB, for example. And I'm just going to go back to the home page, and I'm going to quickly, let's run an RNA-seq search. And I'm going to pick, let's just pick it. It doesn't really matter. I'm just going to pick a, let's take this acute versus chronic infection from Laura Knoll's group. And I'm going to go ahead and take, going to ask for genes that are upregulated during the chronic infection. Let's keep the defaults. And so let's see how many genes come back. You'll see there are 540 genes that are upregulated during chronic infection. And let's say you're interested in potentially using some of these as a vaccine candidate. Well, one thing that's going to be very important is that they don't have orthologs in mammals or in humans, right? Because you don't want to start working on a protein that's potentially the antibodies against that protein would also recognize, or the immune response would also recognize a host protein. So one thing to do is to ask, well, how many of these genes have orthologs in mammals, or don't have orthologs in mammals in this case? So I'm going to add a step. I'm going to run a search, and in this case, I'm going to look for the phyletic pattern search. So again, there's orthology phylogenetic profile search. And so this is the same search available to you in OrthoMCL, but it's available here in any of our component databases. And I can quickly go in here and say, well, out of the metazoa, I want to make sure that it is not, I don't want any sequences that are in mammals, for example. And I'm going to intersect the results. So I'm looking for any sequences that are in that do not have orthologs in mammals, and I'm going to intersect the results with my previous ones. So looking for genes that are chronically infected. I never ran the search, so we're doing an experiment here, basically. I don't know what the results will be, but that's the nice thing about this, is that these experiments, these in silico experiments, are you know, they don't necessarily cost you lab reagents, but and you can actually modify them and play around with them. So you'll see here that 396 of my genes do not have or belong to orthology groups that do not include mammals, right? So these presumably are genes that do not have orthologs in mammals. You can go down and look at them more closely and obviously ask additional questions about these genes. All right, we're at the top of the hour. I think this is all I'm going to cover here. Maybe give people a minute if they have any questions. Suzanne, Eve, and Gloria, anything else you want to add?
Nope,
Nothing
here.
here.
Okay. And as a reminder to everybody, this webinar has been recorded and will be available on YouTube. You should get a link tomorrow when you get the automatically generated certificate from GoToWebinar. If you have any questions, as usual, later on, you can click on the Contact Us option up here. Also, in case you did not notice, there's a community chat option on the bottom here. The goal of this is to make sure that people can chat with each other and ask questions. But you can see here that there are a number of questions that were asked, and there are a number of replies. And so you can actually go in and start asking your questions, and hopefully not only ViewPathDB staff will answer you, but other people as well. All right, well, I think we'll go ahead and end here. Thanks everybody for joining. Enjoy the rest of your week, and we will see you in our next webinar. Thank you, bye-bye.
Bye-bye.
Thank you.
Bye.